Alright, so yesterday we went through the steps of meiosis. So this is a reminder of what it looks like. If we had a cell that had two pairs of chromosomes, and I'm going to draw them as two different colors for a reason. And then this is the second pair of chromosomes. One came from your mom and one came from your dad from each pair. In meiosis 1, I showed you that they would line up side by side and they would go through what's called independent assortment, meaning it's completely random which one of these two goes to the left and which one of these two, second pair, go to the left. Well, let's just say that the two white ones both go to the left in this particular example. It could have been a white and a black. This would be meiosis 1. At the end of meiosis 1, we would have made two cells, and each of those cells would have had the haploid number of chromosomes. So our original cell was 2N, and our two new cells are in, haploid. Meiosis 2 does not cut the chromosome number in half any further. Now, just the X is split in half, just like what happened in mitosis, which makes now 4 cells. Now, yesterday I told you that the number of possible combinations in those 4 cells was 2 to the n, where n is the haploid number or the number of chromosome pairs. But based on the picture here, it doesn't actually look like that's the case, because remember how chromosomes, when they go through S, they make an exact copy of themselves, and the two chromatids, the two sides of one chromosome, are identical. In other words, in theory, the left side and the right side of this chromosome should look exactly alike. And the left side and the right side of the second chromosome should look exactly alike. So based on that information, it would seem like these two, if this was a male, these two sperm would be identical to each other, in theory, because these chromosomes, the left and the right sides, are identical to each other. But that's not the case. Every single gamete is different. And that's because of this last part, crossing over. So I'm going to get rid of this. So what happens in crossing over, and I have a little animation of it afterwards. During meiosis 1, remember meiosis 1 was subdivided into prophase 1, metaphase 1, anaphase 1, telophase 1. In prophase 1, the chromosomes actually start to pair up. But they don't just pair up. What they actually do is sort of wrap around and attach to each other. So if I make the dark chromosome and the light chromosome here, they would literally be sort of wrapped around one another like this. And they actually form a protein lattice that connects them to each other. So the two chromosomes are not just close to each other side by side, they're literally stuck together. And the name of that is synapsis. Now, what happens during synapsis is that they literally, little pieces of each chromosome will break off and switch places. And that's called crossing over. So what we end up with at the end of prophase 1 is it's going to look something like this. Here's my original chromosome on the left. Actually, I'll just do them solid. And here's my chromosome on the right. And the one on the right and the one on the left, little pieces switched places. So now, the two chromatids in a chromosome are not identical. They're actually different from one another because of crossing over. Now, this is going to come back in the genetics chapter, and we're going to talk about how sometimes you'll have traits that are carried on the same chromosome, because you have thousands of traits and you only have 23 different chromosomes. So obviously, every chromosome can't just code for one thing. Chromosomes code for multiple things. So some traits are carried on the same chromosome, and when that's the case, you tend to inherit those traits together, because if you inherit this chromosome, you're going to get every single trait on it. But crossing over allows there to be combinations that didn't exist before, because now this chromosome carries new information instead of the information it originally had, and the same is true of the one on the right. So this is called crossing over. This happens I think it said, I read 1 to 3 times per chromosome pair every time meiosis happens. And it's completely random where the crossing over happens. It can happen in a middle section, like they can overlap each other and swap a middle piece. They can swap a tip. They can swap multiple tips. The information, remember, on the two chromosomes is about the same traits, but it doesn't mean that it's the exact same DNA codes, because this one might have coded for brown eyes before, and this one coded for blue, and now that information has been swapped. And now this chromosome carries, blue, and the other one carries brown. And I have an animation to show this. So this basically further increases the variation. We get new gene combinations that didn't exist before fertilization even happens. Mathematically, you can actually calculate, it's called gene mapping. It comes up in another chapter. We can figure out how far apart genes are on a particular chromosome by how often crossing over happens. The higher the crossover frequency, the further apart the genes are. So we actually know, we actually can predict how far apart. We can map out the locations of genes just by seeing how often we get new combinations. So let me show you this animation of it. And it's probably going to pop up somewhere in the middle. So I'm going to let it finish and then start over. Okay, so this is the original cell. This is the cell before S. This is a cell, it's in G1, let's just say. Now it's going to go through S. All right, now look at crossing over. This is prophase 1 of meiosis. Notice how pieces of the chromosomes, matching chromosomes, their pieces are switching places. At the end of crossing over, you now see these two chromosomes, the two chromatids on one are not the same. Now it's going to go through the rest of meiosis 1, and now meiosis 2, and notice all 4 cells are different. There are no identical cells because of crossing over. And every single time gametes are made, this happens on random chromosomes in random places. So this is the final way that variation is further increased when cells, when we go through sexual reproduction. Question? This would be making, like, say, sperm cells. So 1 pre-sperm making 4 sperm cells. And the same with eggs. Even though you technically only make 1 giant egg, it would still be random which one of those 4. Yes. All right. This is another diagram of it. This is actually showing the place where the crossover is happening. I know they figured a lot of it out through studies, which I'm going to show you some of the experiments in the next chapter of how they discovered a lot of this information. This is just showing 1 pair of chromosomes crossing over, and then the 4 chromosomes in the daughter cells. Again, none of those 4 are identical to each other because of the crossing over. All right, this is just a comparison of mitosis and meiosis. So this is the cell in S before mitosis. And now mitosis happens here. Metaphase, they split, anaphase, and you have 2 cells. The 2 cells in mitosis are 2N, just like the original cell. Or if the original cell was in, they'd be in. But the bottom line is they are identical to the original cell that started the process back in G1 before it went through its cell cycle. Here's meiosis. Here's the DNA making a copy of itself. This is S. Now here's prophase 1. You see crossing over happening. Here they line up, metaphase. They split. This is meiosis 1 here, and then meiosis 2. And now notice how the cells that we've made here are haploid. They have half the chromosome number of our original cell at the top. And we'll review all of this again next week. All right, so last slide. How does sexual reproduction, we know that the benefit of sexual reproduction is that it increases variation, which in the course of evolution has allowed organisms to survive when the environment changed. There are 3 ways sexual reproduction increases variation, and these are the 3 ways. 1, meiosis, the fact that it's completely random which chromosome from each pair ends up in each cell, and that's called independent assortment. Crossing over, that I just showed you, that not only do the chromosomes sort independently, but pieces of them swap places with each other. So you're not even getting a whole chromosome intact. You're getting a chromosome that's been shuffled around. And finally, fertilization. The fact that you now have not only all that stuff happening in one parent, but all that stuff happening in the second parent as well, and then the genes getting together in the offspring.